Hello, Usha ma'am.
Yes.
Yes, ma'am. You can start the introduction.
Good morning, everyone. Today, we are most privileged to have a live lecture by Dr. Sharma, who is the head of the department, microbiology, from Swami Shri Swarupanand Saraswati Mahavidyalaya, Billai. Dr. Sharma is basically a microbiologist and who has served for the past 31 years. And she is very enthusiastic and has served many institutes in the field of microbiology. And she has attended and presented her papers in conferences, seminars, and she published more than 37 research papers and obtained 7 patents. And she has been in editorial board for 7 books. And she is also a NAC coordinator. We welcome you, ma'am, to this stream of webinar series. Ma'am, kindly proceed, ma'am. You can start, ma'am.
Thank you very much, ma'am.
Thank you.
So, the audience today, we will be dealing with the role of microorganisms in biotechnology field. I'll be sharing my PPT.
Today's topic is role of microorganisms in biotechnology field. It would be very useful for all budding microbiologists, research scholars, and faculty members. I welcome all the faculty members, research scholars, and students of this field and related fields of microbiology. We from the on behalf of Microbiology Society, I invite one and all with great pleasure. Thank you all.
Ma'am, is it visible? My screen is visible?
Not yet, ma'am.
I am sharing.
Audience, if you have any doubts, you can put it in chat box at the end of the lecture, and you can also ask. We will unmute you. Please raise your hand once the lecture is finished. Thank you all.
Ma'am, here it is showing that my screen is visible. I have shared the screen.
Not yet, ma'am. You have please click that and share it, ma'am. I think you have selected one folder. Click that,
Yes.
ma'am. Click that, ma'am.
Okay. Now, is it?
Yes, ma'am.
Is it visible?
Yes, ma'am.
Okay.
You can start, ma'am.
Okay. Now, what is the biotechnology or microbial biotechnology? Biotechnology is a field in which the techniques are being used using the microorganisms or the living things. The microbial biotechnology, if we are dealing with the microorganisms, is the application of the principles of biotechnology and the techniques for the utilization of the microorganisms such as bacteria, fungi, and other microorganisms to perform various tasks that are beneficial to human health, industry, and environment. If we see the history, it is a very long history of contributing the development of new products and technologies. It is an area of active research and development with many exciting possibilities for the future. If we see back, the discovery of penicillin in 1940 and in 1940, its large-scale production has led to the mainstream of the biotechnology. And the real bloom in biotechnology started in 1950s, where when the molecular biology and the cell function discovery took place and we came to know about the 3D structure of the DNA was discovered. In 1960s, the synthesis of insulin and the development of vaccines for the diseases like mumps, rubella, and measles. These lead to the golden era of the biotechnology. In 1980s, the development of first biotechnology derived vaccine and drug for cancer and hepatitis was done. And if we see in the history of microbial biotechnology, we will be seeing that the antibiotics which are being produced, hormones, energy sources, food products, and probiotics, these all are the products of biotechnology, and biotechnology has helped in these fields. Proteomics, genomics, drug development, these all involves in microbiological fields. When these all are involved, they lead to the field of biotechnology. There are, if we see, there are 5 types of biotechnology: human biotechnology, environmental biotechnology, industrial biotechnology, animal and plant biotechnology. All these biotechnology are related to a single string, that is the microorganisms. Now, telling about you about the father of biotechnology, Carl Irke, in 1878, is the father of biotechnology. If in India we see, the first biotechnology company was started by Kiran Majumdar Shaw in 1978, and Department of Biology came into existence in 1986. With this brief introduction, I would like to move towards the topic that what are the examples of microbial biotechnology applications. They include the use of bacteria to produce antibiotics and other pharmaceuticals, the use of yeast to produce food and beverages, and the use of fungi to decompose organic matter in landfills. All these includes the uses of microorganisms and its applications in the field of biotechnology. The microbial biotechnology also involves the use of genetically modified organisms to perform a specific task. As I said that genomics leads to helps in the development of genetically modified organisms. And this GMOs, such as the development of crops that are more resistant to pests or that can grow in challenging environments. In this, we can also know about the Bacillus thuringiensis. You can we can talk about the RDT, that is the recombinant DNA technology, which is used to create bioengineered microbes by manipulating the specific functional genes of the significance, which helps in crop pest resistance protection.
Excuse me, ma'am. Dr. Shama, ma'am.
Yes.
Ma'am, PPT is not visible, ma'am.
Not visible?
Yes, ma'am.
Okay. Your screen is sharing. Here it is showing that screen is sharing.
But it's not visible, ma'am. Can you rearrange, ma'am?
Okay,
You,
I am arranging.
Okay.
Is it now visible, ma'am? Now is it visible, ma'am?
Yes, ma'am. Yes, ma'am.
This was the second slide. No,
Yes.
Is it visible?
Yes,
Okay.
ma'am. You can proceed, ma'am. Thank you.
Okay. So these microorganisms or the microorganisms which are derived, bioengineered with the help of recombinant DNA technology by manipulating the specific functional genes of significance, which may lead to the crop pest production, biological waste degradation, increase in biological nitrogen fixation, biofertilization, phytostimulation, salinity, disease control, production of stress-tolerant plants, etc. If we see these RDT technology has made the microbes, bioengineered microbes, which are used in the wastewater treatment, biocorrosion control, biofuels and biodiesel production, disease treatment, nanobiotechnology in the production of various chemicals, pharmaceutical testings, and bioremediation. Now, this era is the modern microbial biotechnology and microbiome, the era of the omics era of microbial biotechnology, which includes the DNA sequencing, genomics, transcriptomics, proteomics, and metabolomics. Modern microbial industries have been developed. Modern medical microbiology, which involves the various microorganisms and alterations in the microorganisms leading to various biotechnological products. Modern food production. In food production also, we use the various microorganisms for the nitrogen, biological nitrogen fixation, as well as as biofertilizers. Modern environmental biotechnology, single-cell proteins, bioinformatics, and microbiome in modern microbial technology. In human health, we use these microorganisms for disease, for the remedies against the diseases, aging, and beauty. The microbiome in the various agricultural systems. In agricultural system, in plant, animal, and food all together, along with the microorganisms, are helpful in making a biotechnological and enhancing the biotechnological field. Earth and environmental microbiome. Am I not audible?
Audience, kindly mute your audio.
Am I not audible, ma'am?
Yes, ma'am. Audience, kindly mute your audio.
Am I audible, ma'am?
Yes, ma'am. Yes,
Okay,
ma'am.
okay. So the microbial biotechnology helps in managing the biotic stress. It helps in managing the abiotic stress also. It promotes the bioremediation. With the help of biotechnology, we alter the gene sequences of the useful microorganisms to promote the bioremediation. It improves the biomass-derived energy techniques for the various energy products. It helps in the value addition in the crops, and which leads to increased productivity and the quality of the crops. And as earlier said, that it is being used for enhancing the nitrogen fixation and increased nutrient uptake with the help of the biofertilizers. Now coming to the main motto, that is what are the microorganisms? From, if we, it last backs from the history, if we see that these microorganisms are used in the production of yeast is being used in the production of wine, bread, etc. And these key industrial microorganisms are Saccharomyces cerevisiae, which is used for brewing and bread making, Escherichia coli for recombinant proteins and others. Aspergillus niger is a fungi for manufacturing citric acid and various enzymes. Clostridium botulinum is a bacteria which is used in the soy milk and cheese. Xanthomonas campestri is the bacteria that produces xanthan gum, and Dinoecious radiurans is a bacteria for soil and water remediation. These key bacteria are then modified and used with the help of techniques like I have made one table which will be telling you that what are the industrial application that depends on the microorganisms for their corresponding products. Food and beverage production, if we will see, the microorganisms which are involved are the yeast, fungi, and bacteria for beer brewing, wine making, and for the manufacturing of the dairy foods like cheese, yogurt, sour cream, cottage cheese, and others. Various microbes are used for the production of the sauerkraut, pickles, soya sauce, kimchi, and other fermented food products, in which comes the probiotics also. The microorganisms are used in the wastewater treatment, and industrial and municipal wastewater can be treated with mixed microbial populations to remove the suspended solids, the dissolved organic compounds, and to oxidize ammonia. With the help of, actually, these microorganisms and biotechnology has a relation that without the help of these microorganisms, we cannot start the process of biotechnology. And when these techniques are being applied in the microorganisms, these microorganisms give their 100% for the production. In bioremediation also, the naturally occurring bacteria also bioremediate, but the altered bacteria, it makes it faster and remove the pollutants such as the polycyclic aromatic hydrocarbons, halogenated hydrocarbons, heavy metals, and others from the soil. They are used as biocatalysis for the production of specific enzymes such as lipases, amylases, DNA polymerases, serine proteases, and others which are used for catalyzing the reactions. These are used for biofuel production. Bacteria, yeast, and algae have been used to manufacture bioethanol and biodiesel as transportation fuels, as well as biomethane and bio-derived hydrocarbon. We can make best out of the waste with the help of these microorganisms by forming the biofuel production. Bio-based chemical production. The engineered bacteria and algae have been used to manufacture organic intermediate chemicals and solvents such as acetone, biobutanol, ethanol, lactic acid, succinic acid, butanediol, propanediol, butadiene, and others, and many more have to be discovered. They are used for the pharmaceutical productions. Bacteria and fungi have been used to produce several classes of antibiotics, antifungals, chemotherapeutic agents, enzymes, proteins, nucleic acids, and vaccines. They are used as biopolymers. A number of polymers such as polyhydroxyalkanoates like PHA, polyesters, polysaccharides, and polyamides are synthesized with the help of microorganisms. Bioleaching. Bacteria have been used to extract metal ions, including gold, copper, or cobalt, from ores, especially low-grade ores, as well as from sludges and wastes. Bioleaching. Microbes have been used to remove lignins from wood pulp in the paper industry. And as food additives, these bacteria and fungi have been used to manufacture a wide range of food additives, such as polyhydric alcohols like mannitol, citric acid, xanthan gum, vitamin C, and other vitamins, as well as they are used for various flavors and colors to be provided. In genetic engineering, the viruses have been used as delivery vectors to insert genetic materials into other cells, and we all know the plasmids are used in this. So, some newly isolated microorganisms. These microorganisms were present, but only we have to isolate them and know the things for which they can be used. Some newly isolated microorganisms are used for aroma, that is for flavor or the smell, as surfactants, for the production of the lipids and the polysaccharides. And the application of these newly isolated microorganisms can be used in food industry, for health and nutrition, in cosmetic formulation, in environmental applications, in bioremediation, in biological activities, and in pharmaceutical and the medical fields. Now, how to proceed with these microorganisms? How will we be proceeding with these microorganisms in the field of biotechnology? So first of all, we have to identify and characterize the beneficial strain from the environmental sample which we are taking. And then we will be seeing it in vitro analysis will be done for these microorganisms, in vivo analysis, GRAS analysis. Its health benefit will be studied, and the GIT survival will be also studied in case of the microorganisms which will be used for the for human beings. GRAS analysis means the generally recognized as safe, the organisms which will be generally recognized as safe for the use of in various fields. After that, when we have identified and characterized this bacterial strain, the second step comes towards the production and the processing. That means the industrial biotechnology. First part is the microbial biotechnology. Second part is the industrial biotechnology, in which we are deal with the production and the processing of the product. In this, what we do, we optimize the method of the production or the standardize the process, and the micro encapsulation is then, and fermentation is being done for the production. After we have produced and processed the product, what we have to do, we have to store it. So for storage, we follow the various treatments such as the chemical treatments and the lyophilization technique. Finally, the team comes for marketing, and then the microorganisms which are being biotechnologically modified and have produced the product are marketed in the for the use of human beings. The process which I have told, that microbial biotechnology, we can know in this way, that how this technique is being used, how this biotechnology is used for the microorganisms. So first of all, what we do is that we collect the sample from the environment for the metagenomics, and we extract. What we do the two parts. First, we extract DNA from those sample, and we extract RNA from the sample. And then we develop a metagenomic library so that we can study it very carefully. And then we sequence this DNA for phylogenetic marker gene. And then we sequence it for metagenomic DNA also. And bioprospecting, when we develop the metagenomic library, what we do, we are bioprospecting it, and for the assessment of taxonomic diversity and for its metabolic potential, and then we classify it that this is a particular microorganisms and what changes can be done in this. With the help of RNA extraction, what we know about its community functions, and we know about its community members, that which type of protein or which type of enzymes they can produce, and that can be classified further and made, developed or changed with the help of various RDT technology. So when we have modified an organism, I am taking an example that for sustainable bacterial cellulose, what we do is that how we modify it. First of all, we use this microorganism with the help of the tissue engineering. We modify it, and after modification of this bacteria, we blend it, we coat it, we dope it, and then we apply it for various processes. And this recycling is done with the help of those bacteria which are being tissue engineered. And like this, the bacterial cellulose get recycled for the various types of productions, which will be helpful in the tissue engineering as well as in the battery for electrical vehicle, for the flexible display, etc. And it leads to the recycling, and we degrade this cellulose with the help of these microorganisms which are genetically engineered. Like this, we can do for various other microorganisms, and for each and every microorganisms, we have to follow the path according to our product which we want there. So it all depends upon the product. And these microorganisms, when they are biotechnologically bioengineered, when they are bioengineered for the useful of cells, they lead to a variety of good products also, and they help us in the sustainable development as well as in removing the pollution and making the earth much more suitable. So thank you very much, all of you, for listening so carefully. And if some or the other type of microorganisms you want to know for this field, you can ask me that what type of help which can be provided. For example, many of the microorganisms which are used in the biotechnology are Azotobacter, Azospirillum, Bacillus, Cyanobacteria, Enterobacter, Gluconobacter, Klebsiella, Herbaspirillum, and Pseudomonas foetida, Rhizobium, Trichoderma, etc. So I think so.
Thank you so much, ma'am, for lucid presentation. The presentation is excellent, ma'am.
Thank you so much,
Audience,
ma'am. I have just
audience,
collected all the microorganisms which are used in the biotechnology, most of the organisms, and the process, how we should proceed for for biotechnology. Actually, the students or somewhere the audience can ask any of the questions which they require.
yeah. We have a few questions in chat box, ma'am.
Okay,
Shall I-
I'll be.
Yeah, how to cultivate wild edible mushroom through biotechnological approaches, ma'am.
I may see in the chat. Okay.
How to cultivate wild edible mushroom
Edible mushroom through
biotechnology
biotechnological
approaches.
approaches.
Yes,
Actually, one thing we should know that the microorganisms which have been identified, or these mushrooms which have been identified, they have another their history is somewhat different, or their life cycle is somewhat different. Halami sir is asking the question. I think so. He is the man who can give me the good answer about this, that how to cultivate the wild edible mushroom through biotechnological approaches. Because sir, at CFTRI, you are doing a lot number of work in this field. My work is not in mushroom, but I'll try to answer it as to cultivate this wild edible mushrooms, we have to change some of the other types of the gene sequence in them, or we have to add some nutrients, because they are wild, they can grow anywhere, but we have to add some good nutrients so that this mushrooms can grow in the desired environment. But sir, I may ask you, it's my request that you can tell us a very good answer to this question. So, Halami sir,
exactly,
please.
Dr. Shama, what you told. Actually, during rainy season, lot of wild
Yes,
mushrooms,
lot of mushrooms are there in our
actually,
garden
just
also.
because my place is almost nearby to Raipur, Chandrapur.
Okay.
And then I was just talking at home, and they said that they prepared the mushroom, which is actually the button mushroom, very small.
Yes.
And that actually grows only in the forest during rainy season.
Sal forest.
Yeah, exactly. So that is what I was just
Yes.
wondering from yesterday. That you will be the right person, I think, to answer or put
No, sir,
student
I am
to
not
work
the
on this
right.
aspect.
I am not working on mushroom, but yes,
Yeah, actually,
I think so.
mushroom,
It is some
yes.
symbiotic relationship, sir.
Yeah,
That
exactly.
mushroom,
Mushroom
I am
science
also
and
thinking like that.
yeah, before I joined CFTI, there was actually a department, Mushroom Science and Technology, but now that department is almost closed.
Yes, sir.
Since we have this Mushroom Institute in Simla.
Okay, yes, in Shimla and in Meghalaya also, sir, Shillong.
Yes. One thing, another thing, no, related to cultivation, they don't do in CFTI, mostly the product development, value addition to the product.
Okay.
So maybe
Thank
we
you,
should,
sir. Prakash, sir.
we should
Very glad
request,
to hear from you.
we should request Dr. Deshmukh. I think we should have one brainstorming exclusively on this aspect also.
Yes, sir. In mushroom,
Definitely.
we would be liking to have some good speakers, so that we can also know the techniques that how to develop. Because now around about 25 years back, we had some labs working on the mushroom, but now none of them are working on the mushroom in Chhattisgarh. But we are having good variety of mushrooms like futu, which is maybe a symbiotic relationship with the sal forest, and then bodo. Sir, one more mushroom is there that is known as bodo.
Okay. So proper identification.
Morals. I think so.
Yes.
They are morals.
Studying their taxonomy and safety is required.
Yes, sir. That is required.
Beside the propagation, then I think
Yes.
it should be possible.
Yes, sir.
Thank you.
Welcome,
Thank
sir.
you, sir. Prakash, sir. Glad to hear from you, sir.
Thank you, ma'am.
I am Dr. R. Usha Sri from Chennai. Ma'am, we have few more questions, ma'am.
Yes, yes.
How waste disposal and
Zal-
pollution can be controlled through biotechnology using
using
microbiology
microbiology.
tools?
Yes, sir. The waste disposal and pollution control can be done with the help of the bacteria, the biotechnically engineered bacteria. We are working over that, madam, that oil spills. We have got 4 to 5 types of bacteria which we have sent for the identification. So these microorganisms, they degrade these pollutants and use them as the nutrition sources, and they degrade it into the smaller particles, which they use for themselves, for the growth of themselves. And in this way, we have to work through it. We have in Ambikapur, Dr. Prashant has created a ball of microorganisms which are cleaning the pond water. The sewage water, the organic content is being converted. They are using this organic content as the food, and the water is having a good potential. After that, the water is getting better and better. So we have to first of all isolate those organisms and use the potential of those microorganisms in the various fields. In our lab, we are doing for oil eating bacteria. Means we are developing the bacteria for this, for the pollution control of the oil and sewage material.
Thank you so much, ma'am. We have one more question from Sidra Noor. Can we
Yes.
pursue industrial biotechnology as masters with BSc in microbiology?
Yes. You masters in microbiology.
Yes, ma'am.
She has, can we
No,
pursue industrial biotechnology?
Masters in industrial biotechnology
Okay.
with BSc in
BSc in
microbiology.
microbiology. Yes. Now it's open for everyone. Like after National Education Policy 2020, we can have this industrial biotechnology, and they can do the biotechnology in MSc after pursuing in MSc, BSc microbiology. You can do it. Sidra Noor, you can do it. Yes, in industrial biotechnology.
Audience, any other questions?
Ma'am. Hello.
Yes,
You can
I will be
unmute.
sharing Anjali
You can
Ranjan.
unmute and ask us.
Anjali Ranjan has said that you want this PPT for revising, and okay, I'll be sharing this PPT.
Ma'am, can you provide your email ID, ma'am, here?
Yes.
So that, if
Yes.
any queries, they can ask you.
Yes, they can ask me. I am providing my email ID. I will provide this PPT also to you.
Yes, ma'am, so that we can also share it in
Yes.
respective groups, MBSI groups, ma'am.
Yes, sir. Yes, ma'am. Surely. Or the students can ask me from the mail also, and I am typing my phone number also. They can contact WhatsApp number.
Audience, the mail ID is available here. You can share it with your friends.
Mail ID and contact number, WhatsApp group number I have shared. So they can have it. If anyone want to ask any question?
Dear
Ma'am.
audience, any questions?
હેલો.
Yes.
સ્વીસ ચીઝ બનાવવા માટે પ્રોપિયોનિક બેક્ટેરિયમ શર્માની બેક્ટેરિયા વપરાય છે. એનો આકાર કેવો હોય છે?
Sir, your question is not clear. Voice is not
स्वीट
clear,
चीज
sir.
जो स्वीट चीज होता है वो बनाने के लिए प्रोपियोनिक बैक्टीरियम शर्मानी यूज होते हैं बैक्टीरिया, तो उनका शेप कैसा होता है?
Okay, okay. That cheese making, for cheese making.
Yes.
Propionibacterium shermani for
Yes,
Swiss
ma'am. He
cheese.
has asked a question, ma'am. Can we use this Propionibacterium for cheese making? That's isn't it? This is a question from the person, ma'am.
Propionibacterium shermani for
Shermani
yes,
for
it can
cheese
be
making.
used.
लेकिन उनका शेप कैसा होता है? बेसिलस
Propionibacterium fredenici is used for making Swiss cheese. Shermani, I think, is a pathogen one. I'm not sure.
No, sir, but it is being used for making cheese also.
Okay, yeah,
Yes, but
actual,
you are saying that it is pathogenic. That is true, that it is pathogenic, sir, but it is being used. I think so. The strain might be different.
Yeah, actually, I worked on Propionibacterium fredenici. This is actually a starter culture for Swiss cheese.
Cheese, yes.
So what he
The
is
same
asking is
question.
correct only, but safety has to be evaluated before that.
Yes, that safety has to be
Yes,
taken. It should become under that DS GRAS, generally recognized as safe.
It should be a grass status culture.
Yes, yes, yes. If it is good coming under FDA and GRAS, we can use it. It is used. Propionibacterium shermani is used for Swiss cheese, but has been reported that because of using this, there are many bowel syndromes also, and different types of syndromes get there.
Okay.
Yeah, so while using anything which are being for the production of human intake, we have to follow the rules of FDA as well as GRAS. We have to check for that also.
Yes, ma'am. Thank you so much. Any other questions, audience, my dear audience?
So thank you, a big thank you to
Thank
all
you.
of.
Shall we wind up, ma'am? Shall we
Yes,
wind
yes.
up? But
Yes,
there are
yes.
no more questions. Thank you, ma'am, for
Thank you so
lucid
much.
presentation
Thank
and
you.
very clear presentation. You explained very well. I
Thank you,
hope
Usha ma'am.
very useful information. We are most privileged to have you, ma'am.
Thanks to MBSI. Thanks
Even
to
in future,
Deshmukh sir.
we would wait for many more lectures like this.
Thanks to
Thank you.
MBSI. Thanks to
Prasad sir,
Deshmukh sir. And thanks to Halami sir also for joining and
yes.
being with us in his tight schedule of work.
Thank you.
He's a very busy man.
Parallelly, I'm doing something actually.
Yes, but you have given me time.
I was noting down and
Oh,
then writing actually, typing.
okay. So thank you so much, sir.
Thank you,
Thank you,
madam.
Usha ma'am, for giving
Thank
me a
you. Thank you.
chance.
Thank
Thanks
you,
to
ma'am.
Thank you,
MBSI
madam.
also.
Welcome.
Thanks to all the
My
audience.
pleasure.
I'll be sharing my PPT also. You can contact me in at my mail ID as well as my WhatsApp number.
Thank you so much, ma'am.
Thank you.
Thank you, ma'am. Have a great weekend. Thank you.
Thank you.